Okay, hi guys, my name's Rob and I'm just going to talk to you a little bit about the environmental drivers of invertebrate functional groups. So just to introduce the topic briefly, you've probably heard quite a lot about insect declines. They're very well reported in the news and the media. Maybe some of you are familiar with the concept of dark diversity, the fact that we don't really have a good understanding of description of a lot of different species. And then the final concept that I'm going to introduce to you is dark ecology, which is we still also don't understand how these insect groups respond to their environment. So Laura did a really good job of introducing the concept of ecosystem function. But the next concept is this idea of functional groups. And essentially these are groups of organisms in a community or an ecosystem that kind of do the same thing. So they could be disease vectors, they could be herbivores, they could be pollinators, they could be involved in nutrient cycling, but they could also be predators and parasites, which kind of mediate all these other effects as well. And the problem is when you study insects is that they're very, very diverse, which means often that you're limited to studying a few groups at a time. And also because of this diversity and how hard they are to work with, we also have a really limited kind of spatial scale in our understanding of their ecology. So normally limited to a very few sites at the same time. But we really know that our functional groups are comprised of a lot of different types of species. And these species are distributed over really, really wide environmental gradients. So we have these biases in our understanding of their ecology, how they respond to their environment and things. And this is really important for addressing things like insect declines and biodiversity conservation, because we need to understand their ecology before we can really kind of begin to protect it. So the IBA project has provided us with a really unique opportunity to kind of understand insect ecology a bit better. First of all, it's really spatially extensive. It's our samples are across really, really wide environmental gradients. We have a really taxonomically representative sample. We're not limited to a few key groups. We have representation from all of the insect groups in Sweden. And then finally, in Sweden, we're really lucky because we actually have our functional groups already assigned to insect groups, which is really nice. And this was due to a bit of work that Frederick has already presented. So now when I'm talking about functional groups, I'm going to be talking about feeding guilds. And what this means is basically just tells you what things insects are eating. So we start off with two groups, phytophages and saprophages. So these are plant feeders, decomposers. We then have predators, which feed on other invertebrates. And then for each of these groups, we also have groups of parasitoids, so these parasitic wasps that lay their eggs on or in. So each of these groups, phytophages, predators, and saprophages, they have their own parasitic communities associated with them. So some of the questions we might be interested in answering about the dynamics of our different functional groups. So the first is how do these groups respond to their environment, first of all? What are they sensitive to? What do they care about? Second is, do these responses differ among these trophic levels? And then finally, what do these dynamics look like in time and space? How are they varying over the country and over time across the period of a year? So a quick bit of methods. I realize this is kind of a mixed audience, so I'm not going to get too technical, but essentially we use some statistics to model guild species richness over time and space. And then in our model, we have a few variables that we might want to account for and control for. So maybe the effect of habitat, maybe we want to include a spatial term in our model to kind of control for spatial sampling biases. But what we're really interested in is evaluating the effect of environment. For the purpose of this talk, I'm going to concentrate on temperature and precipitation, because we know these things are things that insects are really sensitive to. And then the framework we've used is essentially looking at these non-linear effects of temperature and precipitation, because we know that they are, they might have a thermal optima, they might prefer slightly wetter or drier environments. So, just to dive into some results, what are the environmental drivers of invertebrate feeding guilds? So this plot on the left, it will show you the effect of temperature, accumulated temperature, basically how warm an environment has been over the course of the year, and the kind of what you want to focus on is if one of the coloured lines, which represent the guilds, is over the horizontal dash line, it's having a positive effect on species richness. If it's below, it's having a negative effect. And what this graph tells you is basically low temperatures, they will result in low species richness, high temperatures result in higher species richness, but up to a point. They all kind of round off, so we know that they have like a preferred temperature. This is where these guilds are the most kind of like diverse. But when you look at precipitation, all of our guilds are much less sensitive to how wet it is. They do have some kind of like optimal conditions, but in general, they're much more sensitive to temperature than they are to how wet the environment is. So that's kind of interesting, but what do these responses look like across the different trophic levels? So now I'm going to show you a figure that tells you how sensitive hosts and parasitoid communities are to temperature. What you need to focus on is if the line is above the dashed line, hosts are more sensitive. If it's below the line, then the parasitoids are more sensitive. And what you can see for temperature, so our plant feeders and our decomposers, parasitoid communities are much more sensitive to temperature than their hosts are. And this might be interesting for a reason I should show you slightly later, but they might show slightly different dynamics in time. But precipitation, you see slightly different pattern. So for plant feeders and predators, parasitoids are maybe slightly more sensitive to rainfall, but for hosts, for the decomposers, the hosts are more sensitive to rainfall. So the hosts need more rainfall than their parasitoids communities are. Okay, so that's kind of interesting, but what does it mean, like in terms of like what these communities are doing throughout the course of the year? And so this is just a quick video to show you the change in species richness of a guild throughout time with response to temperature. And I'll just show you one video. I think this is for plant feeders, but basically you can see as you go through the year, it gets warmer, species richness increases across the country. Maybe not the most interesting result, like when it's warm, there are more bugs. But what is interesting is how different they are between the parasites and the host communities. So this figure shows you something slightly similar. If the graph is more red, then you have a relatively higher host richness. If it's blue, then you have a relatively higher parasitic community richness. And as you can see at the end of the year, you can see this wave of parasites coming in to eat the hosts throughout the course. But what's even more interesting is the difference between these dynamics between guild communities, like guild pairs, parasitoids and hosts. You can see these waves of parasitoid richness throughout the year. They really differ between each guild community. So for the decomposers, you can see it happens much earlier. This big wave of parasite richness comes throughout the year compared to the phytophages or the predator communities, which is kind of like a really nice, interesting result from this project. So I've thrown quite a lot of information at you, but just to kind of recap. Environmental drivers, very sensitive to temperature, not so much to rainfall. There are big trophic differences between parasites and their host communities. And these dynamics are really, really variable in time and space. So yeah, thanks for listening.